to Know All for GCSE. Today we're going to be looking at Biology Paper 2 exam questions. Now some of you had asked for this video and videos like these for the sciences because you found the Biology Paper 1 video helpful. So if you do want to check that out, you can check out the previous live stream that we did. So let's start with Biology Paper 2 and let's ask the first question. So first question is, compare the structure and mode of action of the two control systems. So what they're saying here is compare the structure and mode of action between both the nervous system and the endocrine system. So let's go to the whiteboard and begin to answer this one. So let's compare the nervous system and the endocrine system. So the nervous system uses nerve cells and neurons, which are the gaps between the nerve cells, to transmit electrochemical messages to the brain, to the CNS system, and the endocrine system. So the nervous system is for reflex actions, mostly. But the endocrine system tends to be for a longer duration effects. And they are from hormones which release via glands. Okay, so the nervous system electrical impulses along the neurons, and they are quick and last a short time. So reflex actions, therefore, they're quick and are short time. And the endocrine system works in optimal conditions. That's another thing you need to say. So it works in optimal conditions. If these conditions are not kept in balance by, optimal conditions, and it's transported within the blood plasma. So you can say hormones released from the glands and it's released into the blood plasma. And it has longer and slower effects. Okay, let's now move on to the next question, which is, explain what this statement means. Okay, so we have to explain how homeostasis maintains these optimal conditions. So let's move on to the next question. So homeostasis is the regulation of internal body conditions. Now, they're not necessarily asking for the definition of homeostasis, but this will help us in answering the question. So it's asking us, what does the statement it means it maintains optimal conditions mean? So we have to think, what are the optimal conditions for? But we know that enzymes require optimal conditions to work, because otherwise they will denature. This then means that homeostasis enables these optimal conditions, such as pH and the temperature, so that the enzymes can work to the best of their ability. And if enzymes denature, you also need to mention how the reactions will then be too slow. Because remember, enzymes don't cause the reactions, it helps the reaction. Okay. Let's move on to the next question. Joanna eats a toxic sweet and feels tingling in her mouth. Salivary glands, comma, describe the pathway of a reflex action. Okay, so we've got the fact that she eats a toxic sweet, a path, and feels a tingling in her mouth. So let's go through the reflex action for this question. So the first thing we get is information is that she eats a toxic sweet. And we know that the toxic sweet is acidic. So the stimulus, which is a change in the environment, is that there is a change in pH to acidic. Now, what detects the stimulus? We know that receptor cells detect the stimulus. In this case, the receptor cells must be on the tongue. Now we have the relay neuron, the motor neuron, and the sensory neuron. We have the sensory neuron, and the sensory neuron transmits electrical messages to the relay neuron. And at the relay neuron, we have the CNS, central nervous system, comprised of the brain and the spinal cord. And then this means that we then have the motor neuron. And the motor neuron then takes it from the CNS to the decision, so the effector. Now, the effector can either be a muscle gland when we're talking about the reflex arc. In this case, what is trying to happen here is we're trying to balance out the pH change of the acid. Therefore, we need to release a chemical. And so we go to this, and the gland in particular is mentioned in the question. It goes to the saliva gland. Now, the saliva gland releases saliva, and this then neutralizes the acid, and the reason why this damages the teeth. Okay, let's move on to the next question, which is this. When ice-cold water is drunk, why does the temperature near the brain decrease? Now, for this question, it's all you have to say is, it often tricks people out, this one. All you have to say here is that when you have the ice-cold water, the blood is then cooled at the stomach. Because what happens is you have the ice-cold water, the water then goes into the stomach, and then your blood is equally cooled at the stomach, and then the blood moves around the body, and in this case to the brain, and cools the temperature near the brain. Okay, let's move on to the next question. The next question. Describe how the brain receives information about light entering the eye. So we have to describe how the brain receives information about light entering the eye. So this again is all to do with the reflex arc that we discussed. So let's go back to that question to answer. So the reflex arc in this case, we've got the stimulus, and here it's the brain is receiving information about light. So the stimulus is that there's light. The receptor cells are the retina cells. The sensory neuron goes down the optic nerve. The relay neuron, it goes to the CNS and the brain, and then it goes to the motor neuron. Light, the thing that changes the size of the pupil, is called the iris. So the information gets sent to the iris, which is a muscle. So the effector is a muscle here, and the action taken, the response taken, which is the final step here, is that the iris constricts, reducing the amount of light entering the eye. Now this could be a 2 marker, this could be a 4 marker, so it all depends, and always make sure you try and include as many steps as you can if you know that it's more than a 2 marker, because they're clearly asking for something of high quality. Okay, so the next question. A functional MRI scanner allows a person to move while the scanner makes images of the person's brain activity. Suggest how the scanner could help to find out more about the brain damage a person has. Okay, so we have this question. We have an MRI scanner, and it allows a person to move while the scanner makes images. So it allows movement, and it then takes pictures of the brain's activity. So that's what we know about MRI scans. How could the scanner help to find more about the brain damage that a person has? Okay, so this is likely to be a high marked question. So let's go through it now. So let's talk about the different things that you can do. So firstly, you can ask the person to carry out different activities. And as you ask them to carry out these different activities, then you can see which part of the brain is active or inactive. Now, as you can see which part of the brain is active or inactive, this then enables you to compare. You can compare this with a person who doesn't have brain damage. So compare what this then enables you to do is that you can see exactly where the damage is, pinpoint, and the MRI scan can then be used. Okay. Let's go back to the questions now. Explain myopia and hyperopia. Remember, these are the two sightedness. So next question. So myopia is short-sightedness, and it's when you're unable to see in distant things because your lens is too short and too thin. So let's look at this now. We've got myopia and we've got hyperopia. So myopia is when you cannot focus on distant objects. The reason why is because the eyeball is too long. Now what you need to do is say the same exact thing for hyperopia, except you'll say you cannot focus on near objects, and you say the eyeball is too short. Now, the things that solve each of these eye problems is a concave lens for myopia and a convex lens for hyperopia. Now, what you have is with myopia's concave lens, it then allows the rays to focus, and then for hyperopia, the convex lens then allows it to focus. So, moving on to the next exam question. So, next exam question is: explain how the person's eye could adjust to form a clearer image of an object that is close. accommodation. Remember, accommodation is when your ciliary muscles and your suspensory ligaments work together. They work together in order to change the shape of the lens so that you can see things and focus on things that are far and things that are close. So when something is far from the person's eye, there's actually less focus needed. Because focus means when it's up close. When it's closer to the person's eye, there's more focus needed. Because there's more aspects that you can see of this. So let's look. So when there's less focus, so we're being asked about the more focused situation in which the object is near the eye. Now what this means is that the ciliary muscles contract, the suspensory ligaments relax, and therefore, overall, the lens thickens, which means, and it gets more convergent, so that's another way of saying like it thickens, and then it allows the light rays to allow the image to focus on the retina. And that is all you have to say for that question. So let's share the questions page again. So the next question is going to be about controls of body temperature. So this is quite a common question. Explain how blood glucose levels change after having a high glucose meal. So I actually changed this question. I was going to do the body temperature one, but I realised that a lot of people do get that one right anyway. So let's go over the harder hormonal ones. So glucose levels after a high glucose meal. So when we have a high glucose meal, what happens is the insulin is then released from the pancreas into the person's blood. And what insulin does is insulin has two effects. The first effect of insulin is that it encourages muscle cells to take up more glucose. So it encourages the person to take up more glucose. And the second thing is that it converts the excess glucose into glycogen. Now, this is a negative feedback loop, which means that the blood glucose levels can also be too low, and there might be too little of glucose. So when there's too little of glucose, what happens is the hormone glucagon is released by the pancreas, and what it does is it encourages the glycogen to convert back into glucose. Now, when we said it encourages muscle cells, another thing you could say is that it also encourages liver cells. So those two things are quite important as well. Move on to the next question. We've read so many questions so far, so we're doing a really good start. So the next question is, describe how the blood is controlled Okay, so ADH is all about water concentration levels and the kidney. So let's go and take a deep dive into what that means. So ADH is released from the pituitary gland. I believe it's into the pancreas. Oh, well, okay. So it's just released released from the pituitary gland into the pancreas. And then what happens is the ADH travels in the bloodstream. It then goes to the kidney tubules. And at the kidney tubules, it increases the tubule's permeability to water. H is causing muscle to absorb more and more of the water, which then enables more water to be kept in, and then less water is used as urine. And if less water is used as urine, that then means that there's more concentration of water in the blood. Okay, I'm going to have a little 50-second break, so just hang on with me for a second. I'll share this tab. Okay, I'm back. I just had to check a few stream issues going on. So, we've talked about ADH. Now, in terms of, let's do another question similar to body temperature. This question is describing why something happens. So this one is, describe why the skin goes red when warm. Okay, so let's describe why the skin goes red. So this is, as we know, called vasodilation. So we're explaining the process of vasodilation. But before we start to explain the process of vasodilation, we need to include something else. Remember, everything has a stimulus. So something is causing you to feel warm, but we don't have to mention that because in the question, it's just asking why the skin goes red. So we have receptors in the hypothalamus. Now, the hypothalamus is an area of the brain that controls the temperature. So if we were to look at the brain, cerebral cortex. Now, the cerebral cortex is responsible for memory, languages, consciousness, all of that. Then we have the cerebellum. The cerebellum is responsible for balance and motion and muscle coordination. And then we have things like the medulla and the hypothalamus. And these control rates and things like that. So we have receptors in the hypothalamus. And these receptors in the hypothalamus, the blood vessels near the skin surface, and then the blood vessels near the skin dilate. So they get thicker. And as they get thicker, this then means that more blood flows really close to the skin. And as blood flows really close to the skin, this allows thermal energy to transfer from the skin to the environment. Okay. Let's move on to the next question. question. We're moving on from of insulin and water and ADH, and we're now going on to a bit about amino acids. So the next one is explain how amino acids that are in excess are excreted safely. Talking about the excretion of amino acids. So let's discuss this now. So amino acids. So where do we get amino acids from? Just a bit of backstory. So when we digest proteins, proteins break down in the process of digestion to amino acids. Now amino acids then break down further, and through this process of it breaking down, deamination, but we call it has been deaminated. So through this process, it then means that ammonia is formed. Now we have ammonia, but if you know anything about ammonia, it's the things that are in stuff like hair dye. Ammonia is actually very toxic. And because it is toxic, it needs to be converted to something else. So it's then converted to urea. Now, the process of deamination actually happens in the liver. So we have the proteins to amino acids, which happens in the stomach. Then we have the amino acids being deaminated into ammonia, which happens in the liver. And then the ammonia then converts to urea. And this then allows safe excretion via the kidneys. So we have those different points there. Now let's move on to the next question. So the next question is about humans and hormones in human reproduction. How does changes in the uterus lining adapted for fertilization? So that question is a bit of grammatical errors, but basically, how does the change in the uterus lining, how is this adapted for fertilization? So through increasing the uterus lining, what we're doing here is that we're providing an area for a fertilized egg to attach to. And this process of the fertilized egg attaching to the uterus lining, this is called implanting. So through increasing the uterus lining, it then provides an area for the fertilized egg to attach to. Now this then, and another thing of the uterus lining is that it actually increases the number of blood vessels. Because if there's more muscle and more tissue, this means there's also more blood vessels, and this increases the amount of nutrients available in those areas. Okay. Let's move on to the next question. Describe how hormones control the menstrual cycle. Okay, this is all about the different ones. Now, I would recommend using a mnemonic to remember this. I used to remember this using the one FOLPS, and I still remember it today, even though I've done my GCSEs long ago. So, let me show you. This is FOLPS. Or just FOLP, I think it was. So we've got FSH, estrogen, luteinizing hormone, and progesterone. So these are all of the hormones involved in the female's menstrual cycle. So let's now go through and look at each one individually. So FSH. FSH is responsible for egg maturation, maturing of the eggs. Now, FSH is released in the pituitary gland. An easy way to remember this is the one which has letters, so FSH and LH. These are both released in the pituitary gland. And then estrogen and progesterone are both released in the ovaries. Okay. So yeah, FSH is responsible for egg maturation. Now let's look at estrogen. So another function of FSH before we move on, actually. So it's egg maturation, and then it also causes the hormone estrogen to be produced. So causes the release. Then we have estrogen. Now estrogen, the function of this is to maintain the uterus lining. Sorry about that. The function of estrogen is to maintain uterus lining. But just how FSH causes the release of estrogen, estrogen inhibits the release of FSH because there's no more FSH necessary. So what happens is we mature the egg, then we're maintaining the uterus lining, and then we have LH. Now LH's responsibility is the eggs get released into the ovaries, and it is stimulated by the estrogen. So another function of estrogen is to inhibit FSH and release LH. So LH is all about egg ovulation, and then we have progesterone, which just releases by itself, and progesterone stops the release of FSH completely and LH completely. And yeah, so you can think of it as the longer ones stop the formation of the short form ones, and the short form ones normally lead on to the longer form ones. Now I'm going to talk a bit about contraception. So there has been one question over the past exam papers that have actually been about contraception, and I'm going to give it to you now. So evaluate the condom and contraceptive implant. So here what we're doing is going to evaluate the different forms of contraception. So we have the barrier method of the condom, and then we have the implant method of the contraceptive, which is a hormonal method. So look at that now. So let's write down all we know about this question. So the condom is a barrier method, as we just said, whereas the implant is a hormonal method. Now, the condom is a barrier method, so it actually prevents against STIs or STDs, whereas implant does not prevent against STDs. In terms of the effectiveness, the condom coming. However, in terms of this, one issue is the fact that it can easily be damaged and fail to work. And the implant does not really fail. So it doesn't fail in doing its job. I think it lasts for roughly 2 to 3 years. And it goes in your arm. So it's injected in your arm. Now, another thing we can say is the condom is more readily available. It's free in loads of NHS clinics, whereas the implant you have to pay for. Now, in terms of the implant, what hormones is it? It's a slow release of progesterone. Now, we know the function of progesterone. It stops a release of FSH, and that's why that hormone is used. Okay, and that's what we can say about these two, really. Now we're moving on to plant hormones. So, after plant hormones, let's see what else we have to do. So, we've got plant hormones, and then sexual and asexual reproduction, DNA in the genome, genetics, evolution, selective breeding, a little bit of that, cloning, speciation, evidence of fossils, bacteria resistance, carbon water cycle, deforestation, factors affecting water security. Okay, so we'll hopefully be able to get through all of these different topics today. Okay, so let's do some plant hormones questions. So the first plant hormone question. How does auxin make a plant grow towards the light? So first of all, we need to know the effect of this, what this question is asking, which is called positive phototropism. So let's look at how positive phototropism works. So how does auxin make... So what you need to say here is the hormone auxin groups... Well, first of all, it's found in the meristematic cells. So at the roots and the shoots. And in this case, we're looking at the importance of it at the shoots. So auxin then groups at the shady sides of the shoots. And this is because of light. So light stimulates it to do this. Then what happens is auxin promotes cell elongation in that area. So wherever it is, auxin promotes cell elongation. So by grouping at the shady sides of the roots of the shoots, they're then promoting cell elongation in those areas. It causes the plant to tilt towards the light source. Which means it grows towards the light source. Okay, perfect. That's how to answer that question. Okay, so the next question will again be about plant hormones, and it will be the germination required practical. Germination required practical. So you tend to be asked a little bit of questions about this, so let's go through it. So what you're going to do is, firstly, you can identify the different variables. But I always suggest doing this at the end because of the revision. When we revise, so we've got dependent, independent, and control. So now let's look at the different things. So first of all, you're going to place cotton wool in 3 petri dishes and soak them in each equal volumes of water. So let's draw this out. So we have 3 petri dishes. Okay, and in each of these 3 petri dishes, we're placing cotton wool in them. Now, for each of them, we're soaking them in equal. So we have a petri dish, cotton wool, which has been soaked in an equal volume of water. We are then placing 10 mustard seeds per dish. The reason why there's so many is because it increases the chance of plants to grow there, and therefore that enables the plants to grow. And then what we're going to do is we're going to leave these 10 mustard seeds per dish to germinate in the warmth. Now, once germinated, what you're going to do, or as it germinates, sorry, in the warmth, you need to add equal volumes of water, however often you do it, to each of them. Then what you're going to do is you're going to place them in, you're going to make sure that there's equal numbers of seedlings. So say if only 4 grow in this one, and then 6 grow in here, and 7 grow in here, you can only have a maximum of 4 in each one. And then what you're going to do is you're going to put 1 in very shady. 1 is going to be full darkness. 1 is going to be partial light, and then partial darkness. And then 1 is going to be full light. So we have the 3 different conditions here. Now what you're going to see is you're going to observe the height each day over 3 consecutive days. So you're going to wait 3 days. And then you're going to observe the height each day, and you can record this. Now, in terms of the results, what would the results then be? So actually, it should be tallest in the darkest conditions because it's trying to reach the light. It should be tallest, but however, it shouldn't be green. It should actually be yellow. The reason why it should be yellow is because it lacks chlorophyll because of the fact that it's lacking photosynthesis. So tall and yellow. Now in the partial, it will then be dark on one side and tall on the other. So this side will try and reach that side, and then the side in the light is just going to grow straight and upward. And then on this one, they're all going to grow straight and upwards. So when we have the one that receives the most, when we have this plant, for example, we know that this side, let's do here, this side is receiving much more light than this side, and that then means that there's an unequal distribution of auxin, the hormone auxin. And because there's an unequal distribution of auxin, it then causes more growth on one side and more cell elongation on the shadier side because there's more auxin in the shadier regions. Okay, now let's look at B6, which is all about sexual and asexual reproduction. So let me just share my tab. Okay, so the first B6 question. Compare the advantages and disadvantages of sexual and asexual reproduction. So we're being asked to look at sexual reproduction and asexual reproduction, look at the differences and see what are advantageous differences or disadvantageous ones. So, first of all, let's look at the differences between them. So, reproduction and asexual reproduction. So, sexual reproduction comes from the fact where there are two parents which give rise to a child that is genetically unique. For asexual, we have one parent and it forms a clone of that other parent, of the parent. So you just get the clone. This means that they're genetically identical. Now, what they're asking you to do here is not really compare the different types of reproduction, but compare the different types of produce from the reproduction. So is it better to have genetically unique, or is it better to have genetically identical? Now, in terms of advantages, let's put advantages in green. So advantages of genetically unique is that there's genetic variation. And genetic variation is a really positive thing. That means that more species, there's a less likely chance that the entire species will all get wiped out by the same thing. Now, what you're doing here is when there's genetic variation, you're increasing the gene pool, and the wider the gene pool is, the better. Another thing is when there's genetic variation, this also enables species to adapt. Species can now adapt. However, if they're just forming clones of their parents, they're not really adapting, they're remaining the same. Okay. Let's now talk about the advantages of asexual reproduction. So asexual produces something that's genetically identical. So it's really good for rapid population increase. You don't have to find a mate. It brings a disadvantage to this one because firstly, it's slower, and secondly, the population increases much slower then. So it's a slower form and you have to find a mate, which takes longer. Now, another advantage of asexual reproduction is that, as we said, a single parent is required, which can be found much more easily. And another thing is that its disadvantages, however, can only be suited to a particular habitat. So it's very specific. Another thing is that there's no genetic variation, so they can all equally be wiped out by the very same disease. Now let's do another question. So, explain sexual reproduction in flowers. A lot of people don't take the time to understand just the basic ones about flowers because it doesn't tend to show up in any of the mainstream YouTube content such as free science lessons. He doesn't really talk too much about female sexual reproduction, but it's definitely within there. So what you see is that the flower makes both the flower makes both the male counterparts and the female counterparts. So the flower gives rise to the male and the female parts. So the male parts are what we call the pollen. And then the female is just an ovum or an ovule, an egg. So the female is more like humans. Now, how did that ovule come about? This comes about through the process of meiosis. Just how in humans meiosis is used for gametes, so is it in flowers. So we have the pollen and the ovule. And now we're being asked to explain the sexual reproduction. So you have the male counterpart and the female counterpart. Now what happens is that the pollen of one flower, so pollen of flower 1, then pollinates the ovule of another one. Let's call this one flower 2. So the pollen of flower 1 pollinates the ovule of flower 2. Now, what happens is, as it pollinates it, it then forms seeds. And because it forms seeds, this then means that these seeds can grow into new plants. This can be really helpful. So let's go through that again. The flower makes the male part, which is the pollen, and also the female part, which is the ovule. And then what happens is these two parts are made by meiosis. And then what happens is the pollen of one flower pollinates the ovule of another flower. It then creates a seed, and this seed is then this seed then grows into another plant. Okay, so let's now move on to DNA and the genome. Explain how a change in one amino acid in an enzyme molecule could stop the enzyme from working. So what the question is saying here is, how can a mutation occur, basically? So let's look at this now. So when there's a change in an amino acid, we know that amino acid is actually just a triplet of bases. So when there's a change in the amino acid, and then we know that the amino acid then forms, in this case, the enzyme, then we know that the enzyme has to have a specific shape to break down a substrate. So an enzyme must have a specific active site. Now, all of these proteins fold in a really particular 3D structure, a unique structure that will only form that specific protein or that specific enzyme. Now, what we're seeing here is that because there's a change in the amino acid, this then means that the enzyme is altered. Now, this 3D unique structure is also known as the active site. So the specific active site is then changed. Now, what does that mean if the active site is changed? That means that the substrate will no longer be complementary to the active site, and the enzyme will be futile against this thing to break down, and therefore the enzyme will no longer work. Okay, so it's now 9 o'clock, so I hope you have found this helpful. If you would like, I can put in the comments down below the biology exam questions that we've gone through. They've been really useful. And just as a summary of what we've gone through, so you know, we've gone through nervous systems, the brain, the eye, proteins, hormones, a menstrual cycle, plant hormones, sexual and asexual reproduction, and we've just introduced ourselves to DNA and the genome. Good luck with your revision. I hope it goes all the best for you. And if you have any more requests for videos or live streams, please do ask me in the chat below. Thank you so much for watching, and see you soon.